Alrighty guys, so today I'm going to show you how to draw a eukaryotic cell. So the first thing I'll start with is drawing the cell membrane or plasma membrane. So I'm just going to draw it a little something like this. There we go. And I'll just label that as plasma membrane. Next I'm going to draw in our nucleus. So it's going to look a little something like this. Obviously, I need to make sure I'm leaving little gaps to indicate the pores that we have in our nuclear membrane. And then we have our chromatin and chromosomes and DNA all inside here. So I'll label that as the nucleus. So if you guys remember from class, you'll probably realize that what comes next is our rough endoplasmic reticulum, or RER. So that extends just off the nucleus there. And I'm just going to draw it something like this. There we go, and making sure you're adding a bunch of dots on it, and these dots represent our ATS ribosomes. So the ATS ribosomes are slightly larger than our 70S, which are found in prokaryotes, and they're just attached to the rough endoplasmic reticulum. We can also have a smooth endoplasmic reticulum as well, and that looks very similar, but just doesn't have the ribosomes attached to it. So I'm going to label that rough endoplasmic reticulum. And then I'm just going to write smooth ER here, just for short. So speaking about ribosomes, the next thing I'm going to put in is just a bunch of dots all around here in our cytoplasm, which indicate the free ribosomes. So there's free ATS ribosomes that are found in the cell. Free ATS ribosomes. There we go. And then we can also label our cytoplasm. Next we can add in our Golgi apparatus. So that looks a little something like this. It has these big bulbous ends. And this is where our vesicles come from. So a little something like that. You can often also draw a few vesicles just on the side of it there. So I'll label both of those. We have our Golgi apparatus. And we also have vesicles. Next we have probably one of the most important parts of the cell, our little powerhouse. And that is our mitochondria. So I represent these by just drawing the outside, kind of like a jelly bean, and then making sure we're adding lines on the inside to represent our cristae. So I'll chuck a couple of these guys in as well. There we go. There's our little powerhouses. Mitochondria. And then lastly, we have our lysosomes. So I'm just going to draw them in here like this, just as some circles. Might colour them in as well. They generally tend to stain a bit darker. There we go. There are our lysosomes. Excellent. And that, guys, is how we draw a eukaryotic cell.